So this is the basic process of how vision interprets light into something the brain can understand. So light goes in through the eye, through the cornea, through the cornea, and through the pupil, and depending on how bright that light is, the iris will either expand or contract in order to either magnify that light or try to restrict it a bit. So light passes through the lens, and the lens, its job is to focus it, which is a process called accommodation. This basically means that the lens changes shape, either widening or shortening, making small adjustments to focus the light. So after the light is filtered in the lens, it goes all the way back to the back of the eye on a surface called the retina. This is the main sensory part of the eye, and it's made up of two types of receptors, rods and cones. The rods pick up less light and color, so they are used for night vision. Rods are these little red ones right here. And cones, the other ones, the little green ones here, are used normally for day vision. The next big process is called phototransduction, when light is actually converted into an electrical signal that the brain can understand. Light passes all the way down here, through all these cells, to the back of the retina, to the rods and cones, and those trigger a chemical reaction in the rods and cones, which moves up through these connections to what are called the bipolar cells, right here, in the middle. The bipolar cells move this reaction all the way up to the ganglion cells, which are right here. And the ganglion cells are all connected at the end and bunch up into something up here called the optic nerve, which all connects and then eventually goes into one single body right around the center of the retina that transmits the data to the brain. However, because the nerve itself in that little spot doesn't have receptor cells on it, it gives a small blind spot called a fovea. Alright, so the path from the eye to the brain first goes through the thalamus, in particular a part of the thalamus called the lateral genticulate body, which has cells called lateral genticulate nuclei. This is where the overall stimulus from the optic nerve is broken down into little components that can be understood. So then after that, all the data goes from the thalamus up to the visual cortex, which is in the occipital lobe in the back of the brain, just above the cerebellum, and the image there is put back together, reconstructed, and finally it's at that point where the image is finally made and projected in your brain, and that's when you finally see something. So then after that, the data is sent up to the parietal lobe where further actions can happen and things can be done.